lipid biosynthesis. It is not the exact reversal of beta oxidation, okay? Which I never did. I don't think I ever did show you the video for beta oxidation. I don't think that in here it will work on the iPad, unfortunately. So, but no, one group of students years ago, whenever we did videos for biochemistry, they actually had a video. It's kind of a plain video, but it's got a very catchy tune for beta oxidation that I've heard multiple people use it to help them memorize the names of the enzymes and stuff like that. If you look at beta oxidation, it used to be one of the very first ones that popped up. They had lots of hits on it. And so, okay, yes, it is not the exact reversal of beta oxidation. So one of the things that we need to keep in mind all has to do with the location. So where did beta oxidation occur versus where does fatty acid synthesis? We're going to primarily focus on fatty acid synthesis and not, well, did a little bit with some of the other lipids, but if you just look at the biosynthesis of endogenous cholesterol, there are so many steps that you could have an entire course probably just on it. So we'll just hit the high notes in that, but for fatty acids, we will cover in great detail. Okay, so we have to think about the location for all of these. So, I kind of gave away the answer to the first one. It's beta oxidation. But then where does fatty acid breakdown occur versus where does fatty acid synthesis occur? Okay, where was fatty acid breakdown? Where did beta oxidation occur? In the big, cell scheme, where does beta oxidation occur? Is it in the cytosol? Is it in the mitochondrial matrix? Where does it occur? Those are usually the two biggies, the areas for metabolic, breakdown in particular. Where does glycolysis occur? Cytosol. Where does gluconeogenesis occur? Asterisk, yes, because it's one of the. It depends on your needs and also at what point in the pathway it occurs. If you're the entire pathway, it goes from the cytosol to there's a little bit of it in the mitochondrial matrix and then it goes back out. What about the urea cycle? It's in and out as well. Remember, it starts initially in the mitochondrial matrix of Watt's cell, hepatocyte, and then it ends it outside of the mitochondrial matrix into the cytosol, of the hepatocyte. So when does beta oxidation occur? It's in the mitochondrial matrix. Where does fatty acid biosynthesis occur? And there was the little asterisk with respect to beta oxidation was if it had some of the specialty bonds. There are some enzymes that are located with the ER. We're going to talk about that, I think, with biosynthesis. But most of this occurs in cytosol. Okay. All right. So I'm going to give you guys a moment. This one's not available yet on my Fire. I did put the suggested problems up for this chapter, but not the PowerPoints. I hadn't actually uploaded them yet to the computer for this semester anyway. Okay. Whenever we talk about the biosynthesis, we are starting in the beginning with acetyl-CoA. Right. So now, the first question that we have to address is what enzyme do you suppose is going to start off the carboxylation event of acetyl-CoA? And if you see here, what we have is we have acetyl-CoA. This is bicarbonate, which we have a lot of bicarbonate around. Where does it come from? That one we covered last semester. I think we talked a little bit about this semester, but especially last semester. Why do we have a lot of bicarbonate around? See anything about it? Take a deep breath. Think about it. What? We're not going to breathe in. Don't. You could die if you had too much. If you had too much.
It's a buffer.
What?
It's a buffer.
It is a buffer. Where did the buffer come from?
It's in our small intestine, right?
Well, it's the one that's used actually for the entire serum. But yeah. Mm-hmm. But where does it come from? Does it originate? What molecule originates bicarbonate? Let's just think about it. If you need to, take deep breaths. What?
Carbon dioxide.
Carbon dioxide. Carbon dioxide with water, remember, and then the whole idea. That's carbonic anhydrase to make bicarbonate. Yeah. Okay. The Bohr effect from last semester. It's carbon dioxide. And that's why if you have too much carbon dioxide, you die. Okay, but it does require ATP. You make a biotin, we've seen biotin before. It does require manganese. It's not magnesium, it's not a typo, it's manganese, and it makes malonyl-CoA. Right? So what class or classes of enzymes could this be? I can think of a couple. What one's probably the most logical? Oh, actually, I hadn't even thought of that one. The synthetase, that's one. I did not thought of synthetase. When we talk about carboxylation, it's carboxylase, okay. But you know, synthetase, if you had said synthetase, I'd give you partial, if not all the credit. This one's carboxylase. And if we're going to add the name, I can't speak today. It's acyl-CoA carboxylase, so that's what we're carboxylating. We've seen biotin do this before. Okay. Biotin, remember, it's really good at adding carboxyl groups. So for carboxylases, sometimes the enzyme is called decarboxylase if they're removing it. Usually with decarboxylases, if they remove it, it's in the form of carbon dioxide. Immediately. Okay. This malonyl-CoA is the starting point then. Okay, so just like the step 0, you can almost think of this as being a step 0. We had step 0, you know, for the citric acid cycle. At this point, we are now going to start adding 2 carbon units to each carbon chain. So this one. It is a spiral, similar to what we've seen with beta oxidation. There's 3 functionalities to this enzyme. Okay, that's why this is really an oversimplification. This little way that they like to draw a little scheme here. 3 things have to occur, okay? Let me see before I tell you wrong. I do have this blown up on the following slides, okay? That way you can look at each one. But we have the biotin carrier protein. You're going to have the biotin carboxylase, and then you have the transcarboxylase portion of the enzyme, the big complex, okay? Like I said, the figure on the right, I do have it separated out to where we can see it more easily. We've seen this kind of thing before. Remember, biotin kind of swings. Sometimes they can call it the arm. Yeah, do they call it the arm? Yeah, the arm. We always call it the arm. So what amino acids kind of covered up, but what amino acid do you suppose it is that's going to be on the biotin carrier protein that's attached to, that holds the biotin in place? If you just look at that little part right there, it's kind of covered up with the word share right now. What amino acid does that look like? Well, the left end of it is biotin.
Lysine.
It's lysine. Okay, remember, lysine is a really long bunch of carbons, and it has that amine on the end. And that amine's a great nucleophile, or can be a great nucleophile. So then that's biotin it's connected to. Okay? Bicarbonate by itself is perfectly happy. Because if you draw it out, in fact, a lot of times in Gen Chem, when you're learning about resonance structures, a lot of times they use bicarbonate as an example. And so that's why, you know, it's relatively stable because of the fact that there's resonance that's possible. So you do have to use the energy of ATP in order just to activate it, to add it to the biotin carrier protein. And then the arm. So then what happens here is that the biotin carboxylase is what's going to add it directly onto the biotin, and it's going to swing on over. Whoop. And then that's where the acyl-CoA is going to come in, and it's going to have that carboxyl group added onto the acyl-CoA. Okay. And so, whoops, didn't mean to do that. I was really hoping it would, well, that's neither here nor there. Okay, so, yes, so then once we've done this, we have malonyl-CoA, and so it's this terminal carboxylate is what's been added on by the transcarboxylase group, and then it's malonyl-CoA that's going to go on and be the starting point. Okay. This enzyme that's coming up is one of my favorites. It's huge. Because, many times we talked about enzyme complexes that differ by organisms, or maybe bacteria does, or if you remember, we haven't done this yet. We'll talk about, how bacteria may do something one way versus humans and yeast or eukaryotes doing something else, and blah, blah, blah. This enzyme complex synthesizes fatty acids in all organisms. Okay. And it, it's huge. So, if I just told you there's an enzyme complex that synthesizes fatty acids, what do you suppose a good name for this enzyme complex would be? Fatty acid? And it's fatty acid synthase. See, its name tells you exactly what it does. It's a repeating 4-step process, but it's having 4 separate enzymes. It's this huge, I've got an example of it coming up in just a moment, protein that has different regions. So it's a big factory. It just literally takes, and it's almost like now you may have seen like pictures of the robotic processes that they have in the lab, where the little robotic arm will kind of pick up the sample and put it from machine to machine to machine. This enzyme's been doing that for eons of time. Okay, so that's what it does. It has a repeating 4-step process. It's a saturated acyl group produced each time. So if you notice, we're only making saturated fatty acids. We're not doing some type of desaturates yet. Okay. The very first step, when it has to happen, is you got to activate the malonyl group. And from organic chemistry, whenever we say you're activating something, what does that mean? Like, what does it mean when we say we have to activate it first? We do that a lot of times with ATP. You're doing like a phosphorylation. You're doing something that's going to make it more reactive of an intermediate. Okay, so you can do things like sulfonation, phosphorylation. Thioesters are great, stuff like that. So that's what's going to be happening. So this is an example of what's going on. Don't worry, we're going to break it. We will break it down. This enzyme. Okay. But I wanted to show you what's going on first. Obviously, they've got this as a little cartoon here, but we see this type of decarboxylation in the past, because remember, this is called a beta keto acid. It's a beta keto acid, right? Because this is the carboxylic acid, and that's the beta keto group. Okay, so we have one part of the enzyme that holds it all in place. Malonyl-CoA is great in the sense that since it's a beta keto acid, you can have the decarboxylation of it. So you decarboxylate it by having the negative charge go down, smack that carbon. That causes this to go out and do the attacking. And what it does is it attacks an acetyl group that's on a neighboring region of the protein. Okay, which we've seen this kind of thing. Technically, if you in organic chemistry, you probably learned it smacks a carbon. You get the tetrahedral intermediate. You get collapse the tetrahedral intermediate, and it kicks off the sulfur in the form of thiolate. And so now we have 2 carbons added onto it, and then we'll talk about each of these steps. It looks kind of like beta oxidation. It's not the exact reversal. We notice that we're going to reduce it, make an alcohol. You dehydrate it in order to make a trans fat. And then it gets reduced once again, and you've got your saturated fat. And then you're going to repeat this process again by adding in another acetyl group, and you go on and on and on. These are very complex systems. And for mammals, it's not only is it just one big complex, it's a single protein. It's a single polypeptide chain. It's massive. I think technically the picture I've got is from pig. I think. Yeah, porcine. All right, so I just wanted to tell you that. So this is what it looks like. I think it looks awesome. And for those who aren't colorblind, but this thing is huge. So they name the different regions. They give them abbreviations, but once it makes sense once you see and once you read like what these all do. Little note: 2 of the domains were not crystallized because presumably they call them disordered. Presumably they move around too much. Okay, and that's the acyl carrier protein, which kind of makes sense. It's the one that kind of goes, the little arm for it. and then the thioesterase portion. Okay, and so it's a single polypeptide. It's a massive in us mammals. Okay, so there are all these different domains. They will be abbreviating these domains from here on out, and so that's why I wanted to show this first. You may want to write these down. Some of them, just by seeing what their names are, you could probably already guess what kind of activity that they're going to do. So if we first start off, there's the part that they call the KS. That stands for beta-keto acyl ACP synthase. Then we have the MAT, which is malonyl acetyl-CoA transferase. The DH region, which is beta-hydroxyacyl carrier protein, which ACP, they don't keep writing out acyl carrier protein, they call it ACP. ACP dehydratase. ER, the ER is enoyl ACP reductase. KR is the beta-keto acyl ACP reductase. ACP is acyl carrier protein. As I mentioned just a moment ago, TE stands for thioesterase. It's only used at the very end, okay, to release it from it. Do you remember in beta oxidation, what was the name of that final, the step 4 that broke it apart that made it down to the 2? It wasn't thioesterase, but it sounded an awful lot like it. It had a special name. What enzyme took 4 carbon containing fatty acids? Or actually, they just knocked off the last 2, and broke it down to the last 2.
Just thiolase?
It's thiolase. Okay, so thiolase is the one that's breaking it down. Thioesterase is actually building it up. Okay. But it's the one that's going to release it at the very end. Acyl carrier protein is the one that, just like it says, it carries acyl groups. All these thioesters are acyl groups. So if we just took this step by step, if you had to guess the KS, which is beta-keto acyl ACP synthase, what do you suppose it's going to be doing? Synthases mean it's what? Synthesizing or building up, and what will it be making, if you want to think it? It's making beta-keto acyl ACP, okay? And so, whereas if we had the malonyl acetyl-CoA transferase, what do you suppose it's going to be doing? It's transferring it between malonyl and acetyl-CoA. So that's why these names make really logical sense. The hardest one, actually, is probably thioesterase, because I've seen people get it confused with thiolase. Okay, because you are making a thioester. It's the only one that has a special name. Okay. So I'll give everybody a moment. Okay, so we're going to start off with the acyl carrier protein portion of it. Once again, remember this is it's only a portion of the entire protein. Remember, the protein is huge, it's massive. I don't want you to think that in us that we have 6 or 7 different enzymes all doing this. It's one big factor. Okay, the ACP portion of it. Okay, and once again, a little side note, bacteria, it's a separate protein, but for us mammals, that's where I'm going to stick to. It's actually just a domain of the entire complex. But you can see that it's actually derived from pantothenic acid. What amino acid does that look like that you suppose that we've seen before? It's going to be important on the ACP. It actually holds the pantothenic acid. On it, at the very end, it's got the thiol. That's the business end, as my old boss, or my old teacher used to call it, the business end. But what holds it on to the ACP? What amino acid is that? Serine. It's serine. Yep. So that's just serine. All right. So pantothenic acid is derived from vitamin B5. This all is coenzyme A. I forgot to ask you the enzyme. And so, what biomolecule does this look an awful lot like? It looks an awful lot like coenzyme A. Okay. But once again, this is the important part. This is the I should say just. It is a thiol. It just minimizes it. So, okay. But this is the business end. So it's a reactive thiol. So that's why you can add to it to make thioesters, just like what we've seen with coenzyme A. So here is the reason why I called it, to me, it looks like gummy worms. See? but they have it drawn. But it's also really cool because it just kind of takes it through and the little gummy worm snake looking thing. And don't worry, each step I've got it blown up to take you through it step by step. Okay, so don't attempt to try to draw this all real quick. Just meander with the gummy worm. All right, so round one in the beginning. Well, actually, technically, it's not the beginning because we've already made Milano Kuwait. So, okay, this is a little bit of a misnomer there. So, before the very first step can occur, you activate the enzyme. Okay, and so what we do is we've got the acyl carrier protein, which is very, it moves around a lot, so it's very functional. What's the word I'm looking for? I can't think of it now. I can't think of the word that normally you would use, like with respect to structures. But it moves around a lot. It has to, because it literally will go from space to space on the gummy worm, for lack of a better word. Okay, so what happens is MAT, portion of it, the Milano acetyl-CoA ACP transferase, is going to transfer this all over. So what we've seen here is, well, actually we've already seen this portion. So we've seen the acetyl-CoA, we have an acetyl-CoA coming in, it's going to kick off the coenzyme A, and that way we have an acetyl group on here. Okay, so now it's activated. The acetyl group is on, and it's ready to go. So, now we take the Milano CoA that we've made from like that step 0, if you wanted to call it that, for lack of a better word. We're going to put it on to the acyl carrier protein. Okay? So now the Milano CoA is on this acyl carrier protein, the one that looks like a long arm. We have our decarboxylation portion, so that comes down. This will swing out and smack the acetyl group that we've charged on, that we activated the enzyme with earlier. Okay, so we smacked it. It'll kick off the free thiol there on the KS. Okay. Next. Oh, and for those of you who are into organic chemistry or remember from organic chemistry, this is called the Claisen condensation, or it's related to the Claisen condensation with decarboxylation. Okay? So now we have this beta keto acetyl group. Okay, and that's why, oh, whoops, sorry. That's how it gets its name, because this is an acetyl group. So that'd be alpha, so that's beta. So that's where it gets its name, from beta keto acyl, okay? Right now it's acetyl. The next round, it would be just a beta keto acyl group because of the fact that it has more than 2 carbons. Just want to point that out. So remember, acetyl means 2, acyl means an indiscriminate number. Okay. So I always want to point this out because people get this name incorrect. Let me put it in a different color. This name right here, and I think it's still that way in the most recent edition of the book too, is incorrect because it can only be butyryl. What does that mean, this part? 4. Okay, so don't name it that because the next round it will have 6. And so it's no longer a butyl group. So that's why you should always just call it beta keto acyl. You can't go wrong there, okay? ACP. Because it's technically only beta keto butyryl the first time, and then it's hexyl second time around, and then it would be octyryl maybe? I don't know. Decyryl and all that kind of stuff. But if you just call it acyl, acyls, I just wanted to point that out. Okay. Any questions so far? So this is all the very first step in order to make it look like the beta keto acyl group. It's still called ACP because it's still attached to the ACP protein. For step 2, are we doing on time? Awesome. Step 2, this is where you get reduction of it. Okay. So, first of all, you should notice the required cofactor. It requires an NADPH, not an NADH, but it's a reductase. It's that beta keto reductase. And once again, throughout, I would call this acyl, because it's only going to be butyryl the first time through. But it's going to reduce it, and whenever it reduces it, now it's reducing this position, so it's going to go from a ketone to an alcohol. So now it's a beta hydroxy acyl ACP. So everywhere where you see the butyl part or butyryl part, you will want to put acyl because butyryl only means 4. Not only that, butyryl is an old name for it. Okay, this is where we're going to stop for today. We'll pick up with it on the next and third step. Are there any questions so far? And we're going to pick up where we left off. This keeps coming. So for the third step is where we're going to be doing the dehydration reaction. Right. And so this one, once again, we're talking about between the alpha and the beta carbons. So it's similar to beta oxidation in that sense. Please know, it doesn't show the stereochemistry there. But once again, you do technically form a trans fat, but it's not leaving the enzyme. So you do not have to worry. So when you're healthy, you don't have to worry about this going and clogging your arteries. So it's not like, you know, the trans fats that you may see in margarine. Okay, so, okay. And so this is done by the DH, the dehydratase. And technically, once again, it's beta hydroxy acyl ACP, so it's a butyryl. Be the acyl 1 hydratase. Or dehydratase, I'm sorry. Dehydratase. Okay. All right. And then the last step of the spiral. Okay. So for here, and that's what I wanted to point out for the spiral versus at the very end when you're done making the fatty acid. Because right now, we've only made one cycle of it. So we went from 2 carbons, ultimately to having 4 carbons. Typically, that's not going to be the final product. You know, your body's going to want, if you're typically, are you making fatty acids whenever you need energy or whenever you're trying to store it?
Storage.
You know, it's for storage. So you're not going to want to store it at 4, but you can store it much larger. Okay. And so that's why this would not be the stopping place. But, so here, once again, we have, and it's going, yes, because this came from the preceding figure, so it's going up instead of down like what we're normally used to seeing. But we have this trans fat, the delta 2, just lets you know between what carbons, so it's between carbons number 2, 1, 2, and 3. Butynoyl, but I just want to point out that it's going to be like an alkene group. And so but would mean that it's a 4. We can just drop that and just call it enoyl to be generic if you wanted to with respect to the enzyme. But trans delta 2 enoyl ACP, we once again use NADPH, and it uses up energy because typically we're doing this for storage. So in order to make it into the saturated fat, and so, or what would be the saturated fatty acyl group? Well, technically, it's not truly a fatty acid yet because it hasn't been released from the enzyme for the fatty acid synthase. Okay, so that's why this is the enoyl reduction. All right. So what happens now? We've made 4. So how are we going to go ahead and make 6? Like to add 2 more carbons, and that's the next step. So that's where, if you wanted to think of this as being, you know, we started here with, technically we start with 2 plus 2, but we started with 4 carbons. Now we're going to want to go ahead and make 6. We're going to go around and go again, make 8, and so on and so forth. Okay, so we want to do the next round. And it's really simple. All that's going to happen is it's going to be transferred from the ACP to the ACP synthase style. Okay, the KS region. This is the transfer group then. Then we can go ahead and have the next, milanoil-CoA come on in order for the reaction to occur. Okay. And so you just do the exact same step, 4 steps. You would translocate it again if you wanted to go ahead and make up to 8, and it just repeats over and over and over and over again. Okay. At what point will it end? Because just like it says in the Bible, there's just a season for everything. So, about to hear another quote. I don't know. Okay. So it reminded me of did you guys ever watch the show Golden Girls? Yes. And there's an episode where, I don't remember the connotation, but where Sophia's like the good book says, you gotta know when to hold them, know when to fold them, know when to walk away, know when to run. And Dorothy's like, Ma, that's Kenny Rogers, not the Bible. So it's just always stuck in my mind, like after literally decades now that is in my mind. And so I was about to say, as the good book says, you gotta know when to hold them and know when to fold them. Okay, to paraphrase Ecclesiastes. All right. So I actually completely copied this quote from the textbook. That for reasons not well understood, it stops at 16. It'll go until 16. Technically, it's 16 carbon-containing saturated fats called palmitate. If it's a carboxylic acid, it's called palmitic acid. And it stops. I don't know if it's just because it's got so big and bulky that the fatty acid synthase just can't handle it anymore. I'm sure that there are lots of theories out there, but it does. It stops. Okay. That's when the thioesterase domain comes in to play. That's the only time that it's going to come into play. Notice it didn't in the preceding 4 steps, or even that 5th translocation step. It has nothing to do with it. It's only during whenever we make palmitate. Let's kick it off. Okay, and all it does is it takes a water. I didn't show you. The water acts like a nucleophile, and it's going to free up the palmitate. So, if you count all of these, since palmitate has 16 carbons, it's going to require 8 acyl-CoAs, 7 ATPs, and 14 NADPHs. It requires a lot of energy, just so that way you can store it for later on. You're going to get more energy than that out of it. We've already talked about how you can make like 32 ATPs, from the breakdown, but it does require some energy in order to store it, or to make it for storage, I should say.
Next one.
Mm-hmm.
Is this like similar to how fat brain pills work? So
Fat what?
Like they interrupt the storage of fat, or they're supposed to do at least.
I'm sorry, it's still not.
Oh, so is this overall pathway is that involved like the fat brain pills and how they're supposed to help you not store fat and whatnot?
I don't know how that they work, or if how highly effective that they are. Some of these may just, it's hard to say if they are true inhibitors, or if some of them may be homeopathic. Some of them technically I think have been outlawed even in the past, because there's so many different weird side effects that have happened. So God only knows. I don't know specifically. I would think that if it's fat burning, that you're going to want to kick up beta oxidation, and it's probably going to start going through be similar to a keto diet, where it starts you to go towards ketoacidosis. Because I think some of the negative side effects for some of those pills and things were essentially the same as diabetic ketoacidosis, I think, for long-term use. But some of them I think are. They used to also sell some that really essentially they were they kind of reminded me of. I don't know what those called, sea monkeys or whatever, where you used to put in water and they would expand, because they get in your stomach and they would expand, essentially, right? And so it made you feel full.
Yeah.
Okay. And so then the idea was you just stopped eating and you felt full longer. Okay. This is just pretty much a review. This right here, we kind of went over this in the past, but it just hammers home, and once again, I'm going to focus only on the animal cells. I would never ask you about plants, given the scope of this class. But it is highly compartmentalized. Typically, fatty acid synthesis is going to occur in the cytosol, whereas beta oxidation occurs in the mitochondrial matrix. Okay, that being said, the ER is the other primary place for the special lipid synthesis. So we'll talk about desaturation, because your body does make certain desaturated fats. Elongation for beyond 16 carbons, like really long fatty acid elongation, and late steroids and phospholipids occur in the ER. And then the mitochondria, what it is, the primary one that we're talking about would be the fatty acid elongation, can also occur in the mitochondria itself. Okay? Yeah, that's what I wanted to do. Oh, and then one thing is, one of the reasons why it's believed is because NADPH is only available in certain compartments, okay, in the cytosol. So therefore, it's not available in the mitochondrial matrix. All right. Oh, actually, I think I forgot to look ahead. So essentially, I answered these questions for us. But it does occur in the cytosol. Once again, we can ignore the plants. Plants do it in the chloroplasts. We have to think about where acetyl-CoA normally is, or primarily is, and how to get it to the cytosol. It's going to actually consume more ATP as well. So then we have to think, because remember, if you think that coenzyme A doesn't transport across membranes well, or certain membranes. So where is there a high concentration of acetyl-CoA typically? It's going to be the mitochondrial matrix. And so, therefore, we've got to find a way to get it out. So we're going to use simple shuttles similar to what we've seen. Okay. There are multiple sources of the acetyl-CoA. And so we've got to get it through that holy of holies to get it back out into the world. It's sort of like the cell's rendition of the Great Commission. I know I'm kind of pushing it for faith integration, but I take it where I can get it. And so it's got to go out into all the world. And so the way it's going to do that is it's actually going to utilize a couple of things that we've seen in the past. First off, it utilizes citrate synthase. Where have we seen citrate synthase before? What metabolic pathway? Citric acid cycle. Don't overthink it. It makes citrate. Citrate has a transporter, and then it can go on out. Okay. The citrate out in the cytosol then has got to be broken down, and I didn't cover this one up, but it's a lyase that's going to break it down from citrate to oxaloacetate and acetyl-CoA. Acetyl-CoA can then go on to do fatty acid synthesis. Oxaloacetate we've seen before, because what are some of the metabolic pathways that we've seen it? Where have we seen oxaloacetate? It is. So what name two? I want you to name, if you just name two of them, that's good. Citric acid cycle and gluconeogenesis. It's also involved sometimes with like with aspartate and all that, metabolism too. All right, but yes, gluconeogenesis. Okay, so we have to take this oxaloacetate. Remember, it cannot go through the holy of holies, so we transform it to malate. Right. On my slides, because when it was on the PowerPoint, then I walked through it step by step. We've already answered that question. Citric acid cycle, yes. It's citrate lyase. Now, so what enzyme goes from oxaloacetate to malate utilizing NADH? Okay, it could be. What else could it be? Or if you got it backwards, it would be malate dehydrogenase. It's actually the same. Remember, we've talked about the cytosolic versus the mitochondrial isoforms, and so it is. It's malate dehydrogenase. Okay. Which we've seen malate dehydrogenase before in these shuttle systems, as well as in citric acid cycle. I was coming up with a blank for the moment there. Okay. Ooh, here we go. So the malate itself can pass through the membrane. We've seen that. Malate then can go on. Whoops, where we go? So the malate comes through. The malate goes on. I know it says and does what. We've kind of already answered this. Malate, there are multiple things it can do. It could either go to citric acid cycle, or if you have lots of energy, then you can even go to gluconeogenesis for the oxaloacetate. Okay, so there are different possibilities there. Oops. However, back in the cytosol, another possibility is we can actually take malate and make pyruvate. And this is one of only 2 ways that our body has to make NADPH. Remember, the NADPH is needed for a couple of things. We have 1 or 2 enzymes, but it didn't really matter if it's NADH versus NADPH. But we actually only have 2 ways to make NADPH. One is the pentose phosphate pathway. The second is malic enzyme. I know it's kind of a generic name, malic enzyme. And so if you need NADPH, you can go from malate to pyruvate, and you can make it that way. Okay? And then that NADPH can then be utilized to build the synthesis, to build fatty acids. Okay. Pyruvate, we've seen before, it goes through the membrane. It's got a pyruvate transporter. Then the pyruvate is right here. What pathway is this right here? The pyruvate to oxaloacetate. What metabolic pathway is that? It's only like 1 step of it, though. That's gluconeogenesis. Oh, we answered that question. It's gluconeogenesis. Sorry. All right, so now the regulation of fatty acids. That's where I wanted to get to. So I'll give you a moment to look this over to see, like, how do we regulate fatty acid synthase? And see, why does that make sense? Good. We can actually start the next one. It doesn't matter to me where we start. I guess it just makes, to me, it makes logical sense to start with citrate. So citrate, if you have a large quantity of citrate, does this mean it's going to activate, or is it going to inhibit this process? It activates it. So it's activating the process from acetyl-CoA to make malonyl-CoA. Why does that make sense? Under what conditions do we have lots of citrate? Pardon? High energy. We've got, it's starting to back up in the citric acid cycle. We don't need any more energy, so let's make fat. Okay? For the rainy days, so to speak. Likewise, insulin activates citrate lyase. Why does that make sense? When do we get insulin? We had a lot of sugar, right? We just eaten that Milky Way or Snickers or whatever. So we've got the sugar, we've got the energy, our energetic needs are being met. So let's build fat. Okay. On the other hand, I'm going to start with epinephrine. What's another common name for epinephrine? Adrenaline. So it inhibits this. Why does that make sense? When do you need... You're running from the bear. Remember, you always run downhill from the bear, not uphill. Okay, you're running from the bear, so why? What are you going to want to do? Why stop making fats? You're going to need the energy. You don't keep making fats. We've got to be breaking you down. We've got to have a bit of oxidation going. Run from the bear. Same thing with glucagon, except you're not running from the bear with glucagon. It's the fact that you haven't eaten. So it's either during times of fasting or in between meals or something like that. And then finally, palmitoyl-CoA, which remember that's just using the jargon from organic chemistry. That's palmitate or palmitoic acid that's attached to the coenzyme A. So if it has a high concentration, it goes back and it actually inhibits this process. Why does that make sense? If you have a lot of palmitate around, palmitoyl-CoA, that high energy or low energy? We have a lot of palmitoyl-CoA around. High, you got lots of fat, so we can just shut this down. So it's feedback inhibition. Okay, but just don't get. I know sometimes when you see that YL-CoA, but no, it looks Greek or scary or something like that. I've seen people get confused. If that just means it's connected to coenzyme A, that YL does. Are there any questions? about any of this. Then we're
Or
gonna.
we just explain what was behind the blue box, or is there something else?
I don't remember what's behind the blue box because I can't, and I don't have it on it, so I don't remember what's behind the blue box. Okay. Sorry. What's behind the blue box? Ignore the man behind the curtain or whatever. Yeah. So, but that's not faith integration. That's Wizard of Oz integration.